Hi, my name's Jim Brody. I'm from the University of California, Irvine Department of Biomedical Engineering. Thanks for coming to my talk. I'm going to be talking about genetic risk scores for breast cancer based on machine learning analysis of chromosomal scale length variation. I have no financial relationships to disclose, and I will not discuss off-label use or investigational use of any in my presentation. Let me start with a problem statement. How well can you predict who will develop cancer later in their life based on germline genetics? What does that mean? Well, I'm talking about taking a sample from someone like a baby, a very young person, and analyzing that sample and predicting what happens to that person 60, 70, 80 years later. Is that even possible? Well, it should be. Why? Because the best current predictors of cancer are family history of cancer. So we should be able to do even better than a simple family history by analyzing germline DNA. But we can't. Why is that? Here's a very simplified version of an association study. We have two groups, the patients with cancer and the patients without cancer. In this case, I've indicated each patient is genotyped at 6 locations, and the values indicated either by a capital letter or a small letter. Then the association study proceeds just looking for differences in the two groups. Where are the significant differences? If you notice, all the E's are different. Patients with the variant of the small e get cancer, while those with the variant of the large e do not. This is how association studies work. One problem with that method is that epistatic interactions are invisible to these association studies. Look at this case. Again, we've got these two groups, patients with cancer, patients without cancer. What's different? Well, here, if you have this combination or this combination, you get that in the patients with cancer. You don't see that either of those patterns in the patients without cancer. But if you do a simple GWAS type analysis, it won't show up. Well, how can we get it to show up? One way is to use something like machine learning, which can detect these combinations. But there's a problem with that, and that there's too many features for machine learning. The typical experiments look like this. You have about 10,000, maybe 20,000 or so different patients. But you have about 1 million different variables up here. And machine learning doesn't work well in those cases. So what can we do? You can do two things. One, try to increase the number of patients. That's expensive, time consuming. Or two, you can try and decrease the number of features. Somehow condense the information in a genome into a smaller number. That's what we're trying to do. The smaller number we're working on is chromosome length. And so what we found is that chromosome length varies from person to person. This plot is a histogram showing the nominal length of chromosome 1 for 10,000 different people in the Uk Biobank dataset. Why does chromosome length vary from person to person? Probably the result of many different structural variations, things like copy number variations, insertions, deletions, and translocations. How can we use this? Well, we can take this problem, which had, remember, 1 million different things going across here, and reduce it to something like 22 different numbers going across here. Now we've got a good system set up for machine learning. We applied this to two different data sets. First, the Tcga dataset. Tcga, the Cancer Genome Atlas. This was a project put together by the National Cancer Institute that had about 11,000 different patients, all with one of 33 different tumor types. For each of these 11,000 different patients, they performed very detailed molecular studies that included characterizing the germline DNA. If you use the Tcga dataset, you may be familiar with molecular characterizations of the primary solid tumor. We, on the other hand, are working exclusively with this, blood-derived normal characterizations. These represent the germline DNA. The second data set we used is the Uk Biobank. This is a very different data set. It consists of almost 500,000 people who contributed samples that were genotyped at about 500,000 different locations across the 23 chromosomes. These people enrolled in the study around 15 years ago. Their health records have been regularly updated since then. Out of this data set, we can identify people with breast cancer. In fact, in the Uk Biobank dataset, there are about 1,500 females with breast cancer. Now, one of the complications, they have both self-reported health information, and in some cases, they take the subject's identification and match it with national health records, in this case with a cancer registry. So we have two columns there, self-reported breast cancer and breast cancer as recorded by a national registry in the UK. We had 1,534 cases where both of these were true. There's plenty of cases where someone self-reports a breast cancer, but it's not recorded in the national registry. So those were our cases, 1,534 cases. And then from that, we set up about 4,300 controls. In the Tcga data set, there are 968 cases of female breast cancer. We also selected 3,715 controls. These are all females and they don't have breast cancer. In the Tcga data set, everyone has some form of a tumor. One of the advantages of the Tcga data is that it is pathologist confirmed. All the data cleaning and manipulation was done in R, and we ended up using a machine learning implemented by this H2o.ai. Here's our results from the Tcga. You can see the ROC curve here. We ended up with an AUC of about 0.75. And our results from the Uk Biobank data set. Here we had slightly higher AUC of 0.81. I've listed here the H2o Automl Leaderboard. So what this does, H2o's AutoML does is test out several different algorithms with different sets of hyperparameters and try to find the best performing algorithm. You can see here that we typically get the high 60s for AUC, but there was one deep learning model that produced an AUC of 0.80 almost. H2o then takes a number of these and combines them into what they call a stacked ensemble, and that produced the best overall AUC of 0.816. In conclusion, I've shown that chromosome scale length variation combined with machine learning offers a powerful new method for predicting who develops breast cancer. If you want more information, you can email me, check my website, or Twitter. We haven't published too much on this yet, but we've got preprints on the preprint servers. Also, you can check my GitHub for some code that'll run the Tcga analysis and you can try it yourself. Thank you.